Hello, everyone, and thank you for joining us. We are honored to have Professor Sanket Goel today. He is a professor in BITSI, BITS Pilani in India, where he is currently the dean and spearheads of research and innovation. He's also a professor of electrical and electronics engineering department at Hyderabad Campus, where he headed the department between 2017 and 2020. Sanket has won awards like ISPS Fellowship, BITS Pilani Best Faculty Award, Fulbright Fellowship, American Electrochemical Society's Best Students Paper Award, and University of Alberta PhD Thesis Award. We are honored to have you here today. Thank you. The floor is yours.
Thank you, Patricia, for inviting me. I hope it's a little early for all of you because what Patricia told me that you guys started late, and thanks for sort of adjusting your time just because of me. So this talk, I'll be going to talk under Distinguished Lecture Program by IEEE Census Council. We can see on the left side, we have this, our institute, BITS Pilani. It's the oldest non-government institute in India. It started 1964, and now we have 6 campuses across India, and of course, one in Dubai. This small snapshot of the campuses you can see on your left side. So today I'll talk about the portable devices, which can be used for different kinds of autonomous self-powered sensing applications. So I just thought to keep a title a little different than what I proposed earlier. So I'll just throw this thing somewhere. Yeah. So, and our lab is called as MEMS Microfluidics and Nano Electronics Lab, as you can see here. Top left, this is our logo. Yeah, let's see. Yeah. So, I mean, there are many trends which you can see. I mean, I will definitely make these slides available with Patricia and others so that you can go through these slides and we can probably spend some time in questions and answers if there is some interest. So you can see that there are a lot of devices which is emerging, especially post-COVID arena. Be it chemiluminescence immunoassay devices, a lot of market which we can see, potential we can see. And with the inception of data analytics, AI, ML, digital twin, IoT, a lot of work is happening. How do we make these devices amenable for the users of the masses so that people can use it on the bedside and without any intervention? And they don't need skilled manpower. You can see this is top right side. This is something which came from our lab. We have developed a very small rupees 400, which means less than $5 device, and which can detect glucose, lactate just from the sweat. That is something which we are talking about, how we can develop such kind of devices. I have few devices and sensors with me here on my desk. I'll try to show you as we move along. You can see this is one patch I want to show you in the beginning. This is something which you must have seen. It cost just 1 rupee in India is like 2 cents. If you talk about US kind of dollars. So you can see that what we have done, we have converted this Band-Aid and then we have added a electrode, which is a textile electrode, and it can sort of suck the sweat and it can measure different kinds of diabetes parameters, especially lactate. So that is something which we are talking about, which the device which can go to the masses and a lot of people can use it. A lot of work is going on in optical sensors. Also, when we talk about developing these simple devices, we have to work on different arenas, especially the materials, how these devices can have very simple, inexpensive, user-friendly materials, and without, of course, losing the physical and chemical, mechanical properties. So graphene is one such material, and the device which I showed you, it has the graphene thread, and which we have sort of woven it using the, one of the printer which we have in our lab. Some of the things which I just keep showing in terms of the stories which I would find out. More stories just to sort of keep the interest intact. Point of care devices had a lot of market, and the idea is that how patients can deal with their own problems themselves and also telemetrize their data to anybody in the world. And of course, try to understand how we can not only do IoT, but also digital twin so that people can sort of control their machines in a remote manner and then do lots of data analytics. So I just thought to bring some numbers, some news, some snapshots sorts in this kind of slide so that we are all interested and motivated during this presentation. Except the direct implication of the human health, one major issue is adulteration, food adulteration. You can see some of the news which came out of India and more so from my own city, Hyderabad. Recently, you can see just last year actually, 4,800 kilogram mangoes were seized, and they're all, were adulterated, and so on and so forth. I just thought to share these images or these news broadcasting so that we can understand there's a need to understand how we can develop these devices, these sensors, and because they have indirect implications to the health and more. The more deeper you go, the more challenges, issues, gaps which we see around us. Um, I just thought to share these issues, and they impact directly or indirectly different organs of our body. You can see some of the organs which are here and how the adult trends and different kinds of unhealthy food will impact our body. So just to give you a kind of a baseline to my presentation, now, in order to make sure that we talk about developing very inexpensive, autonomous, cell-powered devices, we have to work on sensors. As an engineer, we may not be able to predict all these five different senses which are there in our body, but we have to develop an understanding where we can convert one sensory parameter to other sensory parameter. We have to develop a proper transduction mechanism so that we can develop different kinds of sensors. And today I'm more talking about the optical sensors which we have developed. But during the course of action to develop sensors is not a new thing which has just started a few years ago, but people have been working in sensors for ages and ages and ages. And whatever we see, do, eat, it's all sensors from down to dusk. But how do we develop more smarter, more intelligent, more user-friendly sensor? You have to take care of all these things which are mentioned on the periphery of this right side image, be it on developing the new fabrication methods, new materials, new mechanism like electrodes, energy harvesting, integration, storage, and of course, a lot of data analytics which can come from cyber physical system world like AI, ML, and IoT. So all kinds of things have to be done to ensure that we are contemporizing ourselves in terms of developing the new, novel, intelligent sensors. Yeah, so just to give you two more news snippets which came from our lab, there are two most famous newspapers from India, Times of India and Hindu. Both have covered our stories. On the left side, we have developed a small DNA amplification device which can complete the entire bacterial amplification in 15 minutes. And we are working with some of the clinical labs to do the clinical trials and of course commercialize these devices. On the right side again, this is a bacterial infection which we could finish in 15 minutes. So these news have come on a single day and with the same device which can do both bacterial detection, both from the PCR side as well as from the direct detection using the AMR kind of devices. So again, a lot of optical detection has been done to these devices. So I'm sure we all know what is optical biosensors. We can divide into three major categories: bioluminescence, chemiluminescence, electrochemiluminescence. And today I'll be more focusing on electrochemiluminescence, ECL in short, and chemiluminescence, CL in short. And of course, you have to take care of many equipments like photo multiply tube, APDs, avalanche photodiode, and of course, plate reader. This is something which is happening today. A lot of people are using these devices. And of course, people are using CCDs or CMOS camera. CMOS camera we all carry in our pockets. And many applications, as I said, food safety, health monitoring, environmental kind of monitoring. So there are many existing technologies. Spectroscopy, we all do it every day. Any scientist working in broad area of science and technology, they have to use spectroscopy in one way or the other. But there are many other technologies which are available for optical biosensors. But there are many limitations which are mentioned over here. And very obvious limitations. You have to spend time. You have to have a trained manpower, cost. Efficiency is something which is not there. We have to maintain the equipment and so on. Many challenges which you see when you deal with these devices. So I'll be talking, as I said, about two major technologies today: electrochemiluminescence and chemiluminescence. Colorimetric, of course, we have done some work, but I will talk about it maybe in a separate discussions. Maybe I'll start with electrochemiluminescence and we see how we proceed in terms of time, and then we can move towards the chemiluminescence. These are something, a very brief definition of both all these technologies, be it colorimetric, CL, and ECL. So I'll talk about ECL first, and then we see how we move in terms of time. We have around 40 minutes now. So electrochemiluminescence, I mean, we can certainly divide this big word into three major components: electro, chemi, and luminescence. And very obvious definition of these words. Electro, of course, electrical stimulation. Chem is, of course, because of the electrical stimulation, chemical reaction takes place, and which eventually leads to the generation of light. That is a very simple definition of electrochemiluminescence. We have to stimulate, we have to provide some electrical potential to start to ignite the chemical reactions. And of course, we have to kind of capture the light. And that will be the deciding factor of the concentration of a sensor. Many unique features, very sensitive, very selective, and I'll discuss some of the applications or some of the examples of these devices. And we can control external voltage, a lot of a huge linear range which we see with these devices. Interestingly, these devices have two major wavelengths, which I will discuss today. One is between 600 and 700 nanometer, the second one is between 355 to 540 nanometers. So chemistry is very well discussed, very well established, but we have to work on how we can sort of imbibe this chemistry into the device kind of format. Now, in fact, people are also using trying to understand beyond these two wavelengths, so that they're trying to tune the wavelength by some chemistry kind of feature, and we are also working on this aspect so that we can tune the wavelength virtually on any range of wavelength, and we are working with chemists in Australia as well as in India as well. So ECL system, of course, as I said before, that we need to provide voltage, and voltage can be provided in many ways. So you can have a bipolar electrode system, you can have a closed bipolar system, you can have single electrode system. So many different kinds of electrode system which can be sort of leveraged to ensure that the ECL system is completely standalone, and we can just provide an electrical input to start the reaction, and of course, we can get the light output. So you can see that we have tried different ways, including single electrode, bipolar electrode, and both closed and open bipolar electrodes. So I think these numbers I have already presented in the previous slide. So I'll just show you what was the entire system development. What are the constraints, variables which we need to ensure these systems. So you can see that one of the first things is that we have to create electrodes because you have to stimulate the voltage, you have to provide the voltage so that chemical reaction can start taking place. And the moment we provide the electrodes, the voltage to the electrodes, we have to validate them, how the image intensity is proportional to the concentration of the electrode. And that is something I'll be showing you with many examples. And we can compare with the predicted actual values by incorporating some of the machine learning algorithms. And of course, we can throw the signal on the cloud by using the IoT capabilities. You can see that we have not only tested these four analytes, but also many more analytes, including vitamins, neurotransmitters, and so on, which I will discuss down the road. And this is one device which you can see that this is a complete device which has smartphone-based capabilities. And you can see that this device, we can, this device, a newer form is in my hand, actually. You can see that we do not have any electrical wires outside, but we can just press this button. We can place a smartphone here, and internally, smartphone will feed the voltage to this electrode, which you see here. And then we can, based on the analyte concentration, we can analyze this image intensity. So I have got some devices with me so that we can have a little better feeling of the work which we have been working on, actually. And you can see that we have also developed many graphical user interface so that we can do a lot of post analysis and trying to understand what kind of intensity value we can get from a given signal, and then we can find out the unknown concentration as well. So a lot of work which we have been doing, and this is one such initial work which we have done. um, Sorry. But yes, you can see that this is the way the device will work. You can provide the 5 voltage output to the electrode which were recently inserted inside the machine, inside the entire device, and then we can capture the images using the graphical user interface. We have a lot of mechanisms which are inbuilt into this GUI, including the IoT and machine learning capabilities. It will automatically crop the image, and it will automatically tell me the entire intensity pattern vis-a-vis to the pixels which we already have within that image area. So this is the way we can completely automate the entire post processing, and we can find out what is the mean and how the mean is corresponding to the concentration of a given analyte. Like in this case, we are trying to understand the concentration of glucose. And the moment you put the intensity which we are trying to find out, we can find out the glucose concentration. So in this case, of course, little bit manual interventions are there, but down the road we have done more interventions where we can make it completely automated. And also, depending on the application, we can make devices which are based on the polymer, like 3D printed device, which you see in my hand, which can be a paper-based device, which I showed you based on the um, uh, detection using the sweat. And we can make devices paper. This is another device which you see here. This is on a cotton cloth. And you can see that how we have developed the textile electrodes, and we can insert it inside and we can use it for ECL kind of application. So a lot of different kinds of methods and devices which we can use. Like this one, which I'm showing you right now, this is based on the polyamide, and we can enable using the laser, and we can create some graphene patterns. Of course, it's not a single layer graphene, it is a multi-layer graphene, but prime objective is to ensure that we have sufficient electrical conductivity to ignite the reaction. So these are the different value chains which we have created, different entire processes from the design, sensor fabrication, validation, enablement of the machine learning, and of course the development of the prototype. Yeah. So now I will show you a few applications how we have used this kind of devices for different applications. In this case, we have tried to understand the analytes like hydrogen peroxide, dopamine, glucose, xanthine, different kinds of vitamins, and so on. And the entire cost of the device is around $80 kind of thing. And the disposable cassette or disposable sensor, which you see in my hand, it is almost nothing. There's no cost associated, because mostly the cost associated is the manpower cost, I believe. But as such, it's almost like 1 cent or two actually. And that's why we make these sensors disposable, so that we don't really need to sort of invest our time, energy, and efforts to prime them and to sort of clean them properly. So the main cost associated cost is essentially the component cost and to create this device, which is almost like $80 to $85. And we can create the entire graphical user interface. And interestingly, the smartphone not only provides the voltage to the sensor, but also can capture the images and can save the images, can do the image processing, can also do a lot of machine learning algorithm, and also using the graphical user interface can give me the unknown value. So smartphone is such a beautiful device which all of us have. And not only the one smartphone, and you can see this smartphone, which you can see in this image, is like around $100. But we have tried even less expensive smartphone, like $40, which are available online, and all the way till $1,000 kind of smartphone, and they work very, very well actually. And of course, we have to do some play with some electronics smartly, like buck boost converter and so on and so forth, to ensure that the complete flow is streamlined and we can get the output without any user interventions. So this is one flow, which is something like this, where we have tried to create the electrodes on the flexible devices. And essentially, we have developed in-house technology, how we can use the CO2 laser with the optimum doses to create such electrodes. And these electrodes can be used for multiple applications. Like in this case, we have done a lot of optimization in terms of what should be the optimum voltage. For example, in this case, the optimum voltage is 6 volts. That's where we are getting the optimum intensity. And the luminol, some of the constituent which are required to ignite the chemical reaction. Of course, we have to sort of optimize the concentration which we need for different concentration like pH, luminol concentration, and applied voltage. So basically, need to spend a lot of time, but we can inbuild all this data into the graphical user interface and design of experiments so that user know that without starting from scratch, using these optimum values, he or she can run the entire experiment and develop the electrochemiluminescence kind of devices. You can see that in our first set of experiments, which was done almost 5 years ago, that's when we have started ECL almost the time of the COVID. And you can see that we were able to get very nice, repeatable and reproducible graphs for hydrogen peroxide. And of course, we have tried with many real samples like water, like the sweat and bodily parameters and so on and so forth, and works very, very well. And also we have tested the same device, the glucose concentration, and these glucose concentration are within the acceptable limit of human body. And we have been trying to work on many different such analytes. In this case, we have hydrogen peroxide and glucose. And of course, I'll show you in my subsequent slide how other analytes have been used in different kinds of ECL devices and so on and so forth. We have also done the real sample analysis. And likewise, in this case, you can see that how the xanthine has been used. And of course, this does not show the entire range of linear range, but it does show a good amount of linear range from 100 micromolar of xanthine onwards, all the way till 500 micron. So the principle remains the same, but we have tried to use different kinds of electrode. Like for example, in the previous case, we have the bipolar electrode, but in this case, we have the single electrode. Where in this repository, we can inject our analyte, our constituent luminol, and so on, and we can create the entire downstreaming of the ECL base analysis. Xanthine, dopamine, we have tried to understand in this kind of device. And we have also done the stability test, and we have found that these devices, especially these polyimide-based devices, they run very well for many, many, many days. And even if they don't work beyond 7 days, but the cost is so less that we can certainly replace these devices with a fresh lot of devices. Moving on, as I said, that we have not only worked on the flexible polyimide devices, but 3D printed devices. These devices, of course, I don't have the exact 3D printed device which you see in the slide, but it will definitely give a good impression of what we're talking about. So you can see that these devices were fabricated using a dual extruder 3D printer, which cost, less than $1,000 or $800 even now. And we have used normal PLA filament, white color filament on one side, and the graphene embedded PLA filament on the other side. So we have developed these filaments in the lab. We have the filament extruder, and we have done a lot of work in this direction as well to create the conductive filament. And of course, a lot of design optimization, modeling simulation has been done in the background to develop or to finalize these sort of parameters in this kind of devices. And you can see that we have used a photomultiplier tube, not the smartphone, by the way, in this case. And so the idea was that photomultiplier tubes are kind of more sensitive to the desired signal, and to ensure that we were able to go down as much as possible in terms of concentration. A smartphone, of course, has a limitation. We may not really go down less than a certain level. So we thought, why don't we try a photomultiplier tube? Of course, it's quite expensive, but we thought that let's try that and see how we can get the even lower concentration of some of the analytes. So you can see that in this case, choline, another biomarker, which was detected using PMT, and we were able to get much lower concentration of choline, all the way till 5 to 6 micromolars. And also, we have compared it with the smartphone. Smartphone, we were able to get around 30, 35, or even more than that kind of limit of detection. The limit of quantification, LOD was around 3.27, which was almost 3 times than the LOD what we could achieve using a photomultiplier tube. So basically tell us that PMTs are more sensitive, but of course, the cost factor is always there. But depending on the user's requirement, we can select whether we're going to use PMT or smartphone or any other kind of photo detection kind of devices, including photodiodes and so on. Now, we thought that so far we have been trying to understand one analyte in one time, but how about we can develop a device where we can use several different kinds of analytes? Of course, the one which I'm going to show you here is slightly different. It also has five different reservoirs where I can use five different kinds of analytes. But in this case, you can see six different analytes which can be used. By the way, one limitation so far we are trying to address how we can uh, sense the mixture of analytes, kind of a simultaneous detection rather than multiplex detection. That challenge we are trying to address even now. But we have done some work in this direction. I can show you some results later on. But we have tried to develop this device, of course, 3D printed and using a dual extruder 3D printer. You can see that what we have done, we have tried to inject different concentration of glucose in different wells of this 3D printed device. And we can easily see that how the different wells with different concentration give me a different color intensity. And this color intensity is an indicator of different concentration of glucose, which is evident from our graphical user interface. And we could try to find out the real concentration of the analyte, which we could find out in any of the wells, any of the desired wells. Of course, this may not be really have a real implication in the bench side, but it gives a lot of confidence that we can do a lot of different kinds of analyte in one go, if not the mixture of analytes. Moving on, we have also tried many more fancy and inexpensive electrodes. Like in this case, we have tried the pencil, simple HB pencil, which are available in market in India. It costs like 5 cents actually, not even 5 cents per pencil. And one pencil again can make multiple electrodes. So you can imagine that the cost of a single electrode is quite less. And we have tried to understand how we can create a 3D printed jig, and we can sort of house these two graphite electrodes on both the sides. And we can use the space in between as a reservoir to inject my luminol, S2O2, or any analyte combination. So we have tried to understand this device to sense the cholesterol. And as you can see that we have used the HP pencil. And we could find out the cholesterol was giving a very nice linear range from 50 microliter all the way till 1000 microliter with a limit of detection of around 16 micromolar. So it gives us a lot of further trust and confidence that even less expensive devices, less expensive electro, which are available around us in everybody's shelf, everybody's pocket, can also be used for ECL detection. We don't need to do any fabrication. We can also create a simple housing. We don't even need to have a housing made of 3D printing. So I just thought to share that how a simple devices can be used. Further down, we have also developed some devices out of paper. I'm not sure that I have this device with me, but we have used a Whatman filter paper. Those of you who have done some chemistry work, you can appreciate that Whatman filter paper. Everybody uses it. So we use different grades of paper, particularly Whatman filter grade 1 filter paper. And to create a sacrificial layer, we had a fire retardant spray, and then we shine a UV light, and we have even tried blue light or CO2 laser and so on and so forth, and they work very well. Of course, we have to tune the dosing and other parameters of the laser, but you can see that we can create the reduced graphene oxide kind of pattern on Whatman filter paper. It's a real device actually. I don't have the real replica with me right now, but you can see that we can create all these kinds of electrodes. And I also would like to emphasize that our aim is not to have very fancy material and going down deeper to understand the material characteristics, but primarily and predominantly, we should have the sufficient amount of conductivity. And in this case, I believe the conductivity was around 1000 Siemens per meter and that sort of range, actually. So what we have done, we have tried to test this device, which was paper. And paper is very interesting. It has the inbuilt capillaries, so that if you inject an analyte somewhere in the hydrophobic zone, it remains there, and it can create a nice reservoir, nice volume of the fluid, which can last for quite bit of amount actually. In this case, again, we have tried glucose and lactate, and it works very well, and it gives us a lot of good range as well as the limit of detection, as we have seen in the previous devices, and likewise lactate detection. We have also tried this both glucose and lactate using the interference kind of analysis. Like in this case, we have tried to understand how the lactate detection will be very, very specific, even if we add other co-reactant like vitamin C, galactase, glucose, xylose, and you can see that this blue kind of graph is related to the lactate, and it does not really change the ECL intensity pattern, even if I mix other analytes. And we have also seen that we could see the stability for more than 10 days for this kind of devices. So just to summarize whatever we have shown you so far, we have tried to understand the devices made out of the polyimide. We call it laser-induced graphene. We have tried to understand different devices using the 3D printed devices, 3D printed material and filaments. And of course, we have tried to understand devices made out of paper as well as simple HP pencil kind of thing. So moving on, we have done much more studies because a lot of gaps which we were able to find out. And the gaps was there. How do we develop the turnkey kind of device? A user press the button and everything is done automatically. In order to do that, we have to do a lot of data analysis, a lot of data capturing. But as an experimentalist, we have limitations to do experimental work in the lab. So we have tried to understand how we can use the machine learning algorithm in conjunction with the smartphone based analysis. So we have used different kinds of like laser induced graphene on polyimide, 3D printing. We have also used the photolithography in clean room, where we have developed this ITO. I think ITO. Oh, this is not ITO, by the way. This is the copper electrode, which was there on a PCB, FR4 PCB. Of course, they're not transparent. So we have also used a simple PCB and trying to etch away different parts of the of copper, and only this part of the copper will remain. And we can create a reservoir using a acrylic sheet, and we can bond it on top of it, so that this is an empty reservoir where we can inject our analyte and other co-reactants. We have also tried ITO device. This is ITO, and we can see these are some of the transparent electrode which we can see. And idea of these electrodes is that we can try to understand 4 different analytes in one go. We can inject one luminol or co-reactants from here, and we can inject 4 different analytes here. Of course, still simultaneous detection is a challenge which we are trying to understand in the future work. You can see this is the carbon PDMS device. We have tried to develop these carbon reservoirs or carbon colonies using screen printing methods. And this is the silver PDMS paste. We have tried to inject the silver using the inkjet printing. So we have tried different kinds of devices, different kinds of colonies, different kinds of framework, and then trying to understand that this capability can be used for different sets of applications using electrochemiluminescence. Um, Again, we have tried to use the same device which I showed you before to understand how biolights can be detected. I think I can probably skip it. And now, I don't have that. So yeah, this is the same replica which you see here. Of course, this is white, this is black, but this replica is something which we have right now. We can press the button and everything is bingo. We don't need to do anything except pressing the button. And we can you can see this. There is a shutter here. Um, yeah, and we can inject our ECL sensor, whether it is a polymer based or textile based or paper based here, and then that's it. Smartphone will sort of internally provide the voltage. Smartphone will capture the images, do the image processing, and give me the data. And we have done a lot of analysis in terms of developing graphical user interface and different kinds of machine learning algorithms have been built in. So this you can see that how the machine learning algorithm and entire graphical user interface is built in to capture the images, to analyze them, to post-process them, to predict what kind of unknown concentration can be measured using the predictive analysis. And all this has been done using the graphical user interface. You can see that I will be a little fast in that way. And then we can do all kinds of analysis using this device. Now, more work has to be done because still a lot of gaps are there in terms of making it a kind of a bedside sample to answer system. So then what we have thought that why don't we try to avoid the smartphone as much as possible. But challenge is that it will enhance our cost. You can see this is one device which we have developed. It does not have any smartphone, but rather it has the Raspberry Pi kind of monitor and other microprocessors. And we can inject our device, our sensor from here and start the device, and it will give you the entire flow through process to detect the unknown concentration. So that is something, a version 2 of the device which we have developed. Um, and of course, we have tried to understand the 4 different analytes, how they will sense and how they can give me the unknown samples, unknown analysis using this kind of device. We have tried to understand hydrogen peroxide, lactate, glucose, and uric acid with these kind of devices. Of course, it shows us very all the sensory parameters, including repeatability, reproducibility, specificity. We have done real-sample analysis. We have also done 30 days analysis. And this device, now it is in TRL4 kind of stage, but we are working with some of the clinicians and trying to understand if we can do a lot of clinical trials and try to see if we can increase the TRL to 6 to 7. Last but not the least, now you can see that the same device is sitting in this form now. We have a much smaller Raspberry Pi kind of interface, and much smaller. If I compare both the devices, almost half of the size of the previous device. But we can use a much more multiplexing kind of feature in the bigger device, which cannot use in this device. Now, this is the most recent development. We have also developed a wearable device. I think it costs around $12 to $13 if you compare. What we can do, it has an electrode underneath. It can suck the sweat from the body and from the skin, and it can tell me that what kind of ECL signal, ECL signal corresponding to analyte, which we can see. So now this is completely ready, and we are working with a couple of wearable devices industry in India, and of course in Hyderabad. We have also tested with artificial sweat, 8 hours battery life, depending on the battery which we would like to use. And of course, it has the cloud connectivity. It can directly work with the Android app, and we can do onboard analysis of the electrode. So different devices which I showed you here in order to develop ECL kind of platform. So we are not just limiting ourselves to bench research and also to sense many analyze, but also trying to understand how we can make the device completely integrated, automated, self-powered, and any user can certainly use it based on his or her convenience. So this is how the entire value chain which we have, coming from the bench research to the wearable device which we have in ECL kind of domain. So this is about the story about electrochemiluminescence. Sorry, I was a little faster than I was expecting, but I just thought to give you the entire life cycle over the last 5-6 years, how we have started and where we are right now. And now we are working with some of the wearable device watch companies, how we can sort of integrate the ECL capabilities into the wearable watches, smart watches, and they can not only do all different kinds of parameters, and they come with anyway with many parametric amplifications or applications, but they can also do a lot of parametric analysis, which can be done by sucking the sweat or other bodily fluids. Now, I will spend a few minutes on electrochemiluminescence. I have 15 minutes left. Probably I'll try to finish in 10 minutes so that we can spend some time in discussions. So electrochemiluminescence, we have a culprit called electro. We have to provide electrical input. But in chemiluminescence, inherent chemical reactions will give you the light. There is no business of electrical input. The chemical reaction itself will give you the light. So there are a lot of naturally available uh, chemical reactions which are available around us, which can lead to the light. And one of the most famous reaction is the firefly luciferase, and it comes in the enzymatic form also these days. So it is a bioluminescence kind of behavior, which can tell me that it's a naturally available luminescence. But the chemiluminescence is exactly like a bioluminescence, but we mimic it in the lab. We do the chemical reaction in the lab, and we can create different kinds of platform using the chemiluminescence kind of behavior. And I'll show you next 5 minutes what kind of work one person can do to create the point of care sample to answer kind of platform. So it is affordable on-site detection system. Some of the virtues and specifications of these devices are mentioned here. They're portable. I can show you the version 1. It looks little unprofessional, but it works very well. In fact, you can see that we have MM11 again 3D printed casing, and you can see that we have an input and output, and there is a slot where we can sort of insert the smartphone, and we have a magnet smartphone can house nicely. And we have used them for multiple applications, including uric acid detection, vitamin detection, so forth, so on and so forth. And in this case, we have tried to understand how we can detect primarily the uric acid, that these numbers belong to the uric acid. So these devices are affordable, less than 2,000 rupees, which is like around $25 kind of thing. And very environmental friendly, and we can get the output in milliseconds actually. So this is the first version which I showed you just now. Exactly what I have in my hand, but of course it carries the smartphone, and we can get the entire readout using the smartphone. And the smartphone can also provide the readout to the computer, to external monitor, and so on and so forth. As you can see that we have also tried to understand how the hydrogen peroxide can be measured in real milk samples. And interestingly, we have also tried to understand how the heating can impact my chemiluminescence signal. And we have seen that we have a 250%, like almost 2.5 times enhancement of chemiluminescence signal when we do the on-site heating. That's why we have an external periphery, which you can see on the left side, and we have a heater underneath so that we can enhance the chemiluminescence signal. So what you see here, the concentration of hydrogen peroxide in milk and the chemiluminescent intensity, very nice linear graph, which we can see in different ranges of chemiluminescence. And we have tried to understand, if we dilute milk, what kind of hydrogen peroxide which we can measure. And it is very much in order of the recovery percentage, as well as the the percentage which we could see from the other benchtop chemiluminescence devices. So just to give you a video of these devices very quickly. So you can see that this is the device which I showed you just now. So it has the inlet and outlet, and of course there's a smartphone which is built into it. Um, and smartphone either can be housed on top of it, or we can keep it separately actually, depending on our requirement. And there is a small, we use papers actually, papers, and we have a wax boundary so that we have a nice hydrophobic environment. We can place the wax, the paper strip, paper reservoir inside it, close it. And we have also tried to, as I said before, try to see if we can change the temperature, if we can compensate the temperature, how the overall sensitivity can be enhanced using these devices. So multiple challenges which we try to understand, and multiple issues which we try to solve. Like, for example, we have tried to enhance the sensitivity using the reduced graphene oxide by 14 times, using the laser-induced graphene by more than 5 times, and the copper electrode used on the PCB almost 9 times. So that's kind of numbers which we were able to see if we do the different kinds of materials which we can use in place of the conventional carbon or AgCl. So in this case, we have also used the antioxidant in green tea samples, which has a huge susceptibility in terms of the chemiluminescence kind of signal. And we were able to understand how different electrode materials can sort of enhance my overall chemiluminescence signal. So what we have done is that we have tried to understand different brand of tea, how the catechin, which is a important constituent in tea samples, it works like an antioxidant in different tea samples, how it will vary when we change the catechin concentration in Lipton tea, as well as the organic tea, which is sort of procured from India. Some problem. Yeah. Moving on, we have tried to develop different versions of these devices. This is the third V3 of this device, which can completely work on a smartphone. And it has a unique feature where we can inject a desired amount of volume of the light And we have tried to understand how we can detect alkaline phosphatase using this device. Some chemistry which is here. And you can see that alkaline phosphatase again has a very nice linear relationship with the concentration. This is the last version of the device, which I will show you here. Interestingly, the device is there in my hand actually. And you can see that it can sort of do the chemiluminescent reaction in 96 well plate. We can nicely house the 96 well plate, and we can multiplex our reactions. We can do many different kinds of testings in one go. And this is my reservoir, which is somewhere here. This is a design actually, and I can keep my paper pad here, and which has the PEX boundaries, and I can try to understand how my analyte can be detected using these devices. In this case, we have tried to understand the malathion, the pesticide, which is predominantly available, and we have tried to understand how concentration of malathion will be related to the chemiluminescence kind of intensity. Yeah, I mean, we have done comparison with the colorimetry work. Again, this is our own work, which I did not show today. As you can see that in colorimetry, we were getting LOD of around 2.35 ppm, but in this case, we're getting almost 20 times less, or at least 15 times less LOD when we try to understand the chemiluminescence kind of devices. So these are the comparison of 3, 4 different kinds of devices. I just missed telling you that we have made this spring kind of arrangement, which can sort of inject a desired amount of volume. This is another novelty which we have to face. Rather than using the bulky pumps, peristaltic or syringe pump, we have made this kind of arrangements. Yeah, I mean, this is about two major technology. I will just spend 2, 3 minutes to talk about our lab, our people, our students who have done the entire work which I have presented to you during the last 45 to 50 minutes. So MEMS, microfluidics, and nanoelectronics lab, we primarily work on two major aspects, sensors and energy harvesters. And the idea is that if we can sort of integrate sensors and energy harvesters, we can sort of deploy our devices in any subterranean, hilly, or remote areas where power may not be available, clean power may not be available in a non-interrupting manner. So we try to integrate them. So in terms of sensor, we work in different kinds of sensors. And the list you can see here, whether it is bio or agriculture or organ on a chip or pesticide detection, water monitoring, environmental parameter monitoring. As an engineer, we focus more on how we can convert one kind of sensor to another kind of output, whether it is in the form of electrical and optical. So input certainly does not really matter, but we try to understand how we can convert input to the output. And in terms of energy harvester, which I did not show you today, we work on fuel cells, solar, a lot of flexible membristors, supercapacitors, and so on. And you can see that we are not focusing on a huge power output, huge energy harvesting, but small energy harvesting in the range of microwatt per centimeter square kind of thing, so that they can be integrated and we can create these standalone devices. To fabricate these devices, very simple methods. I showed some of the methods in my presentation today, whether it is 3D printing, laser writing, inkjet writing, and all the devices from paper to textile to polymer to glass to silicon. And in terms of the output, both optical and electrical are being done in our lab. And we're also doing a lot of work in AI, ML, and IoT as well. So, well, this is our journey. We started 10 years ago, and now we are where we are right now. This is some of the intellectual outcomes from our lab. I'll just skip it. And right now, our lab has three major, or our team has three major labs. One is the MEMS lab, which you see these three pictures here. And the idea is that how do we create and develop the devices using the rapid prototyping, do a lot of modeling, simulation, and benchtop characterization and detection. The second lab is the wind lab, wearable, implantable, and nearable devices lab. A lot of work with the textile, polymer kind of work, where we can integrate it with the sensor system kind of parameter, and a lot of NFCs, a lot of circuitry integration, which we use to create these devices. The third lab is majorly the printed electronics lab, where we do different kinds of printed electronics, be it inkjet printing or screen printing or bio 3D printer Now we're going to hire roll-to-roll manufacturing system somewhere in the lab, which will be housed in the lab actually. So all these things which we try to understand in these three labs. Currently, the lab has many different kinds of equipments to do the both benchtop and automated sensing when we create these kinds of platform. And as you can see that we have embroidery machine, we have inkjet printer and different characterization tools which are required um, during the entire process. We have a clean room, both class 100 and class 1000. And we can do the entire processing down to a fraction of a micron kind of thing. And people use this thing for initial bench top work and initial back of the envelope kind of work. A lot of characterization tools which are there as central facility. And we are supported by many national and international agencies. And we really thank them for their kind support. Without that support, it won't be possible to implement different activities. We have a lot of visitors from different uh, uh, organizations, academia, industry, and of course the government. And we've been publishing in many journals, and our lab has published 5 books in different domains of signal processing, electrochemical devices, microfluidics, 3D printing, and the recent one on the right side, it's a MEMS practical lab manual, uh, which has almost 20 experiments and the entire workflow, entire protocol, which is there for all these 20 experiments. More than that, we have a website on the Wiley website actually. So every experiment has a video and which are housed on this website. So it's a very interesting project which we have completed a couple of months back. Our work has been captured in media extensively. Last but not the least, we have 3 spin-offs from the lab working in different areas of sensors. Cleom primarily works on developing the affordable diagnostic devices. Pyrome works mainly on the optical detection devices and try to understand how the small devices can be fabricated to synthesize different kinds of nanoparticles and quantum dots. Sensom primarily works on agriculture, food, environmental monitoring devices. And these all these 3 startups are very well funded, especially the curriculum, which has won many prizes. Our team, around 7 8 faculty colleagues from different departments. Currently, we are collaborating with many academicians as well as industry people within India and outside India. Our team has around 30 PhD scholars and 5 postdocs, and around 25 to 26 PhDs have already been graduated and working different parts of the world. Our alumni, really proud of them. And our people have won many awards. And this is the last slide. This is the team. I think this picture was taken around 3 months ago, very close to where our MEMS lab is housed. And yeah, thank you so much for your attention. And I'll be happy to answer the question. Before I conclude, I just thought to share with you on the bottom. This is the kind of a conjoined replica of different campuses which we have. The one on the center is a Pilani. The one right next to it on the left is Hyderabad, where I am housed. On the right is Goa, and then Dubai and Mumbai. And we're going to have another campus almost 300 kilometers or 200 miles from Hyderabad by next year. Thank you so much once again. Really appreciate it.
Thank you. It was a quite impressive talk, not only about the solutions and the sensors that you are developing, but also regarding your lab and everything. Any questions from the audience?
Quick question. Thank you very much, Dr. Can you hear me?
Yes.
Yeah, please.
Yeah, my name is Jerome Hong. I'm a faculty of mechanical engineering of UMN. Very nice meeting you. Very impressive talk. So I'm interested in the cell phone-based sensors. You got a lot of cell phone-based sensors for ECL signals. So my question is about how do you control the exposure time of the cell phone sensors? I mean, according to the sample changes, I think that's exposure time would affecting the luminescent signals you obtained, right? And so how do you make adjustment and calibrate this automatically for different the samples recorded under different conditions? Or you have
A very,
standardized
very,
approach to
yeah, very
pre-process
good question.
samples.
And
Yeah.
I agree with you that exposure time can play an important role. One thing is there, cell phone comes with a 30 frames per second kind of thing. So we take images and then we try to do the deep learning analysis to ensure that we capture the same frame in all the analytes, all the concentration of analyte. A lot of post processing and deep learning is involved during the image capturing and of course post processing. So I think this is done using the deep learning analysis which students have done, and we ensure that we take the same frame across all the uh, excuse me, all the concentration which we are studying.
So you did use the intensity of that, luminescence to quantify the analytes, right?
Yes.
So that intensity, how do you adjust that intensity automatically to when the sample has a different level of concentration, for example, of electrolytes, and how do you adjust that to ensure you can compare quantitatively across different samples?
Yeah, I just said before is that we take the same frame rate actually, or same frame across all the images, and the post analysis is done. I mean, of course, we have a person who is expert in deep learning who has done all these things. So I'm not able to answer your question completely right now, but probably we can discuss offline, and I can
Yeah.
try to find out the answer from our image expert, and then probably he can share with you his thoughts.
Yeah, another question I have is you use the same device that for different analytes, right? So are you looking at just the intensity or looking at also the pattern and time evolution of this signal,
No, we are
variation?
using only the intensity, but that's the only chemical output which we are seeing. Pattern certainly does not have any impact on it. We have tried, I agree with you, we have tried, I know the answer to this question, and we have tried multiple thoughts about it, how the pattern can sort of be an indicator to it, but intensity is the only, but luminescence is something which is the output of the chemical reaction.
Okay, so but how do you differentiate different type of analytes, right? If they all just use intensity values, and for different, are you changing the
Yeah. So that is why I know I'm not claiming so far. In fact, we have some recent results which I'm not showing it today. That's why this device cannot really have a cocktail of the analytes. It cannot detect these simultaneously multiple analyte which are mixed in a cocktail. So it cannot do that right now. But
Mhm.
we're trying to understand because we are trying to do multiple wavelength analysis right now. We have started some work recently, so that's why we have to sort of separate different analytes in different wells, and that's how we can find, because we know that A1, A2, A3, A4, A5 here right now. So we can measure
Okay,
the concentration of individual analyte, but we cannot do it in a cocktail of analyte, right?
I see, I see. All right, thanks.
Thank you. I was also going to ask the same question, is like, how do you confirm the selectivity? Because it's like, yes, I know that you cannot do the mixture, but how do you say like it's selective and you are really measuring what you are supposed to measure?
Yeah, I think we have done, Patricia. This question is extremely important whenever you want to design any sensor, and I fully agree with you. We have done selectivity analysis in many different kinds of sensors, actually. For example, I'm not sure whether I'm still, um, I can still share my screen, but maybe I can quickly do that. I hope you can see my screen again. So in this case, the bottom right side, we can see that we have done the analysis of lactate, which is there in one of the wells, which I showed you just now. So it works very well actually, because because it's all enzymatic reactions, by the way. Maybe I missed that point. So in this case, we have lactate oxidase actually. So it has to give you the signal based on the lactate. So each well is responsible to one kind of analyte, and precisely the chemistry is driven for that particular analyte. So that's why it is very, very specific and for that particular analyte.
Totally makes sense. Thank you. Any other questions?
I do have another quick question. So you did mention about you use a bacterial infection detection with your device, but bacterial infection detection, I think you mentioned in one of the slides, and you didn't talk about it in your presentation. So I'm just curious about what is the technology, how it has been evolved. If you can
Yeah, I
provide a
mean,
very quick
that
summary.
work, that work I did not show it because it is still not published actually.
Okay.
Actually, the process is similar. It's just that we have to create an environment where we can complete the entire ECL reaction. So, so bacterial infection, and what we have done is that we have also added the antibiotic into it. So it's basically a AMR reaction. So, so the moment you put the antibiotic, it basically stop the growth of the bacteria. And we try to understand how the ECL signal degrades when we sort of increase the concentration of antibiotic. And we have tried several different kinds of antibiotic, trying to understand that how the ECL signal degrades with the concentration. In this case, so basic concentration and signal, it goes down.
Have you done any work that using microfluidics? It looks like a lot of them are paper or other type of sensors. I'm just curious whether you have done any work just like cell phone integrated with the microfluidics or for flow cytometry or the cytometry type of work. You know, like where you
Yeah, we
have the
have not.
flow that moving, through the system.
Yeah, we have not done cytometry, but we have done certainly microfluidics kind of work significantly, actually, like this one. You can see that a lot of microfluidic channels are there, and then we try to understand the flow itself is one of the major parameters to augment the intensity output. We have done different kinds of microfluidic devices here. Like in this case, we have used these mixing zones also within the PDMS device and try to understand how the mixing and the way they are mixing, and we have done a lot of modeling also in this kind
So,
of
so in these systems are, what is your pumping mechanism? Are you using your peristaltic pump or using some pressure pump? It's a very small device. I'm just curious.
Yeah, that's the downside of it. We have used the peristaltic pump in this case actually, but again, the point which we're trying to make here is that like different devices can have a different uses. Like for example, this device where we have the pumps and so on can be used in the primary healthcare setting. It may not be used in the bedside or sample to answer kind of devices. So different devices we're trying to, but these devices can be used where you need a more precise and a more quick kind of reaction, the output where doctors can take the decision. But some of the sample to answer and bedside devices are on a regular testing by the patient itself, so that they give a sufficient alarm on a regular basis, whether they really need to visit a doctor and so on. So I think everything has a positives and negatives. So we have used the peristaltic pump. Right now, we are developing a very small pressure pump actually. But still the problem is that the cost is extremely high actually.
Mhm.
So again, it's a trade-off. What we're trying to understand in what setting and what is the
Mhm.
user, who is the user. So depending on that, we can decide what kind of devices which we are going to bring into the market.
All right. Thank you very much.
Thank you.
Thank you so
That's
much.
quite
Really
impressive.
appreciate your questions. Yes.
Any
Really,
other?
we can discuss offline, Jerome. Yes.
Yeah, I think the work is very interesting. So, my area is mainly in the imaging. So we do a lot of imaging based work.
Oh,
Yeah, so cell imaging, holographic imaging. I don't know whether you heard about that before.
yes.
Kind of label-free
Oh,
optical imaging approach, combine the holographic imaging with deep learning. And so we usually mainly looking at the cells, cells, bacteria, stuff like that.
okay. I see. Wonderful. Yeah, nice.
Any other questions? I think that's it. Great. Again, many thanks. It was quite impressive. And considering the time difference, I'm really thankful that you put all the efforts. And I think it's like 10 p.m. there. So thank you a lot again.
Thank you. Thank you, everyone. And Patricia, thank you so much for giving me this opportunity. Hope to see you soon.
Thank you. Take
Thank
care.
you. Take care.
Bye.
Thanks.
Bye.
Bye.